In this video, we are going to discuss how mRNA vaccines work. First, we'll put the mRNA vaccines in context by reviewing different types of vaccines out there. Remember, all of these vaccines aim to activate your immune system to be able to identify and fight against a particular pathogen once your body is exposed to it. How they go about that is a little different. There are live attenuated vaccines. These contained a weakened form of an actual pathogen that causes a disease. So they offer great long immune responses, but they have some limitations because they contain a small part of the live virus. Examples of these would be the chickenpox virus or MMR. Next, there are inactivated viruses, and these your body is exposed to the dead pathogen. Examples would be polio or rabies. Another common type is the subunit vaccines, and these expose you to a piece of the pathogen. Examples are the HPV or the shingles vaccine. And there are toxoid vaccines that don't expose you to the pathogen itself, but to a toxin or a byproduct made by the germ. Finally, there are a newer classification of vaccines, the mRNA or the DNA vaccines. These were first used in animals in the 1990s and have been studied in humans with cancer treatment since 1992. Scientists started developing mRNA vaccines in response to SARS in the early 2000s and Ebola in 2012, but the work was stopped when those infections were contained. So while we haven't needed these vaccines, and so none have been approved until now, they've been being studied and administered in clinical trials for many years. To understand the technology behind mRNA vaccines, we have to do a quick refresher of protein production. So if we start with DNA, that goes through transcription and is rewritten into RNA and ultimately messenger RNA. This messenger RNA, or mRNA, is exported into the cytoplasm of the cell and provides the instructions to make a specific protein. So keep that mRNA information in mind as we look at how these vaccines are developed. We've all probably heard of the spike protein that is on the surface of the SARS-CoV-2 virus, which causes COVID-19. This is the protein that binds to our cells and gets the virus into our cells. Well, the scientists worked backwards and they found the mRNA that provides instructions on how to build the spike protein. They couldn't inject this directly into us because mRNA can't cross our cell membranes. So instead, they put it into a lipid nanoparticle. Basically, think of this lipid nanoparticle as the envelope that they put the spike protein mRNA into to transport it into our cells. And that is what makes up the vaccine. Once injected into our body, the lipid nanoparticle fuses with our cell membrane and releases that mRNA into our cell's cytoplasm. Remember, the mRNA does its job in the cytoplasm of the cell, so it doesn't enter our nucleus where our DNA is housed. Inside the cytoplasm of our cells, the mRNA does its job and gives instructions to our own ribosomes to start making spike proteins. These proteins get expressed on our cell membrane. When the spike protein shape is expressed on our cell membrane, they attract our immune system cells, specifically T helper cells, that then become activated and release cytokines. Cytokines tell our B cells to proliferate and differentiate into plasma cells that then make antibodies that can identify and fight against the spike protein. The cytokines also tell T cells to proliferate and make memory T cells. So if you're exposed to the virus in the future, you already have antibodies and activated immune system cells that are prepped and ready to neutralize and destroy the virus. This puts your body at a huge advantage as compared to being exposed to the virus in an unvaccinated host. If you're unvaccinated, your body has to identify this new pathogen, get ready to fight it, learn what antibodies to create, and then start producing them. SARS-CoV-2 virus can replicate very quickly. So in the time that your body is doing all this, you can start getting sick and your immune system might not be able to have a quick response. However, after you're vaccinated, if the virus enters your body, your immune system is able to begin fighting it immediately and go directly into an immune response. We talked about mutant versions of COVID-19 in the efficacy part 2 video, but I want to bring it up again here because now that we've talked about the science behind the vaccine, I think it'll make more sense. The SARS-CoV-2 virus needs the spike protein to bind to and get into your cells, and the virus has one spike gene, which the mRNA included in the vaccine is derived from. So while there may be different genotypes of COVID-19, it's unlikely it would mutate enough that an immune response to the spike protein wouldn't be protective. The brilliance of the mRNA technology is that it doesn't contain a pathogen or a pathogen's proteins, but it provides instructions for your own body to create the spike protein to learn to identify it and defeat it. It's a relatively simple process to create these vaccines with taking an identified mRNA with a protective shell to deliver it, and your body does the work. In the unexpected case that we did have a new variant that we didn't have protection against, scientists could rapidly identify the mRNA and create a new vaccine within 6 weeks. As far as safety, these vaccines are not infectious, are not made from a virus itself. They've been studied and used in mRNA technologies and have found few adverse events and mild to moderate side effects, and the clinical trials have shown that they are highly efficacious. The drawback of these types of vaccines is that the mRNA is extremely fragile, so it requires really specific cold temperatures to maintain integrity through transportation and storage. We've received questions about what exactly is in the vaccine, so here's a list of ingredients in Moderna's vaccine. The main ingredient is the mRNA, and then there's ingredients that make up the lipid nanoparticle, and the rest of the ingredients work to keep the vaccine stable and maintain its pH until it's injected. And the Pfizer vaccine has similar components, the mRNA, the components of the lipid nanoparticle, and then ingredients used to keep the vaccine stable. Both of these vaccines were developed using genetic sequencing, and neither was developed using fetal cells, which is another myth that's out there.